coming from Ebola, which now has therapeutic options and a vaccine, I think fungi, in a way, are the last frontier. Very few therapeutic options. We have a lot of scary cases of emerging Candida auris that you might have seen in the news. So I'm very happy to introduce the first speaker, Anders Näär, who's a professor here. And he has kind of been, ahead of the curve in trying to find novel therapeutics for fungal diseases and studying mechanistically what kind of targets could be amenable to intervention. So welcome, Anders. We're looking forward to your talk.
Thanks, Julia. And thank you all for coming here. Great session on Ebola leading in here. It's going to be quite a bit of a change of pace. So as Julia said, Ebola, while there are a lot of therapeutic options have been developed, with fungi, we're kind of on the other side of the curve where we had very efficacious therapeutics. However, there's resistance developing to these options, and we're running out of time in developing new antifungals that can counter emerging species such as Candida auris that Julia referred to. So again, it's a bit of a change of pace. So I'm going to put the last slide first, just to introduce what I'm going to talk about, to get you on the same page. And I hope you all had coffee, because this is going to be quite a molecular talk with a lot of data, and I will try to lead you through all the pieces of data carefully. So, and hopefully, it's a bit of a journey, and hopefully I don't lose you on the way. So what I'm going to talk about today is transcription factor. These are proteins binding to DNA, switching on genes, okay? And the genes here that we're looking at are drug efflux pumps. So they pump drugs out of cells. And that's germane to not only fungal disease, but all the way from bacteria to us. We all have these types of pumps that can pump out drugs from the cells to prevent toxicity, toxic accumulation of drugs. And I'm going to describe the discovery of a mechanism whereby fungi control these pumps. So they're inducible, they can be switched on by the drugs that are being pumped out. So it's a sensory system. So these proteins sit in the nucleus, binds to DNA, and then when these antifungals come into the cells, they bind directly to these proteins, which change conformation. And then they can talk to the transcriptional apparatus, if you will, coactivators and RNA polymerase II, the machinery that helps to read these genes and to promote mRNA synthesis and then ultimately the components of these pumps. And so we were thinking that perhaps by understanding this pathway in great detail, down to molecular detail, atomic detail, if you will, perhaps we can interfere with this pathway and switch off these pumps. They're activated in multi-drug resistant fungi. So that's basically the gist. And I'm going to talk about two different stories that have emerged over the last 13 years or so, and then end with some unpublished exciting data on drug discovery, if you will. So I think most of us think about fungi as a nuisance, something that you deal with sometimes on a monthly basis or not. There are other types of diseases such as UTIs and other diseases that also are caused by fungal infections. And these are diseases that afflict all of us. However, immunocompromised individuals, they can actually acquire other more serious fungal infections, Aspergillus, which is a mold, invasive Candida infections, and Cryptococcus. And these can get into the bloodstream and cause really severe life-threatening infections, especially in immunocompromised individuals, which I will get to in a moment. So so what are these types of immunocompromised individuals? Well, we have the HIV AIDS patients, for example. They lack a complete immune system. Cancer chemotherapy also causes suppression of immunity. It kills the immune cells. Organ transplants patients, they are also on immunosuppressive drugs to prevent rejection of the organs. Finally, there are immunomodulatory drugs in various settings, including in cancer immunotherapy. Underlying serious disease, if you're infected with some or afflicted with some serious disease such as cardiovascular disease or something, you're in the hospital and bedridden, you're also immunocompromised in many cases. Infants don't have a fully developed immune system, and the elderly have a depleted immune system. All of these populations are sensitive to fungal infections. And importantly for this talk, many of these fungi can actually get into the bloodstream, and that has a very high mortality risk, so up to 50% mortality. So there's a lot of interest in trying to find efficacious therapies for these types of fungal infections. Now, there is the challenge because we have very few options. So one of the most commonly used antifungal fluconazole was developed now over 30 years, or 20 or 30 years ago in the 1990s. And this has been exceedingly used in the world, but now there's resistance emerging worldwide to fluconazole and many of the other class members of the triazoles. Polyenes such as Efficacy B and new drugs such as echinocandins, caspofungin, etc., or IV drugs. You reserve those for the most sick patients and in the clinic. So they're not widely used, kind of in the last stage of disease. So you have 3 different drugs. They all target the cell surface or the cell wall synthesis. And resistance is emerging to all of these different drugs. So there's an urgent need to find novel antifungals. So what are the drivers of antifungal resistance? So you can see here both in plants and in humans, resistance to azoles, for example, have emerged in synchrony over the last 20 or 30 years. And we think that this is actually due to a number of different factors that act in confluence. This movement of people, there are mutations emerging in the bugs themselves. There's also drugs are treated for a very long period of time in the very sick patients, and so the bugs develop resistance. As population has increased, there's increased density and increased movement and growth. And so we think that that facilitates spreading of resistant bugs. So among the fungi that I'm going to talk about today are the Candida species. So the most prevalent of the Candida species is the C. albicans. This is a diploid fungus that can have both a coccoid conformation as well as a hyphal conformation. They are probably representing about 70% of infections in the clinic today. They have low intrinsic resistance to antifungals, but they can develop resistance over time, and this is something seen in the clinic. They are also able to form biofilms on catheters, for example, that can be very difficult to get rid of. Over the last 30 years or so, there's been other Candida species rising in the clinic. For example, Candida glabrata, which was almost absent clinically 30 years ago, now represents about 20% of all fungal infections in the clinic. And this is probably due to the fact that it has intrinsic resistance to azoles. That's the standard of care for fungal infections. And this has really become an emerging threat in disseminated infections, so that is in the bloodstream, where they can have a lethality of up to 50%. So, and this is the fungus I'm going to talk about mostly today, about the mechanism of multidrug resistance in Candida glabrata, which again represents about 20% of infections. Now, as Julia mentioned, there's a new bad bug out there, Candida auris. So Candida auris was first identified in Japan in 2009 as an ear canal infection, hence the name Auris. And it has spread quite rapidly from 4 different locations. So Japan, India, South America, and in South Africa as well. So 4 different clades, and rising rapidly throughout the world. And why are we so worried about this bug? Well, it has high intrinsic antifungal resistance. So here's the spread to the US. So it started in New York maybe less than 10 years ago and exploded throughout the US, and now we have 1,000 cases, and most of them, almost all, are azole resistant. So they're resistant to the standard of care. Many of them also resistant to Efficacy B, and some are resistant to the last standard of care, echinocandins. So this is the rise of the drug-resistant Candida auris. This is what we're really scared about. This could be like, maybe like Ebola early on. So it's something that we've really been worrying about. The rise of a drug-resistant Candida that cannot be treated at all. And so actually yesterday, the CDC published a study showing that there are 3 cases now identified in New York area, and I think in Chicago was the third one, showing that Candida auris, 3 cases, they have resistance to all 3 classes of antifungals. So this is a huge concern because there's nothing else out there. Okay, so how are we going to address this? So this is basically the story again that I'm going to talk about. So my lab traditionally has been working on trying to understand how genes are switched on and off over many years. Ever since I was a postdoc here at UC Berkeley, then at Harvard, and now back here again as a professor. And we've been motivated to try to understand how transcription factors talk to transcription apparatus and switch on genes. And I'm going to tell you again about this fungal transcription factor, PD-L1, which is a sentry transcription factor binding to antifungals, switching on target genes that efflux these antifungals. So what are the mechanisms of multidrug resistance? So all the way down to bacteria, it's been long known that the mechanism of multidrug resistance can be, at least in part, mediated through these types of drug efflux pumps, many different types. And a lot of these are actually controlled transcriptionally by transcription factors that sit inside the cells waiting for a signal. And then when this signal, in case of the TET repressor here, a classic example of regulation of these drug efflux pumps, where TET binds to the TET repressor, this is now released, liberating or de-repress this gene, which can now be expressed, and you get pumping out of the same ligand, tetracycline. And this turns out to be conserved as a mechanism in humans and in fungi. So this is a highly conserved function, perhaps by not necessarily evolutionary conservation, but an adaptation that's in parallel throughout biology, if you will. So in humans, for example, multidrug resistance is a big concern in cancers, and that's oftentimes mediated again by drug efflux pumps, such as P-glycoprotein, MDR1. In fungi, as I'm going to talk about in Candida species, we have analogous ATP-dependent efflux pumps, CDR1 and CDR2, that function similarly. How are these controlled? So in mammals, it's been extensively studied. You have transcription factors called PXR, pregnant X receptor. These are members of the nuclear hormone receptor family, like estrogen receptor, progesterone, steroid hormone receptors that you may know about. These are the classic steroid receptors that can bind to ligands with very high specificity and switch on target genes to elicit very specific functions. This, by contrast, is a nuclear receptor that binds with a very low affinity to many different types of ligands, xenobiotics. So different types of toxins coming into the cell that then switches on these drug efflux pumps, as well as cytochrome P450 detoxification enzymes in the liver, for example, that leads then to efflux or detoxification of compounds. So this is a well-known pathway, and people have figured out exactly how it works. So PXR binds to ligands that cause a conformational change, so you can now recruit what's known as transcriptional co-activators. These are proteins that help transcription factors to talk to transcriptional apparatus to help them switch on target genes. What about fungi? So little was known how fungi switch on these types of efflux pumps. We hypothesized that perhaps there's a class of fungal transcription factors called the yeast zinc cluster transcription factors that could represent such a family of ligand-responsive transcription factors. So already known that these factors are very similar to nuclear hormone receptors. They have a negative regulatory domain that responds to small molecules, and structurally they look very similar to nuclear receptors, but they're not evolutionarily related by sequence and structure. So it was already known that there is a gene in yeast and in Candida glabrata. So yeast, I'm referring to yeast as the Saccharomyces cerevisiae, baker's yeast, or which you make beer with. And so that's kind of the workhorse that everybody studies in the fungal field, in the basic science area. And so we've been interested in trying to understand how this transcription factor switches on these target genes. Can it bind to ligands like PXR that I just showed you? Can it interact with transcription co-activators to facilitate the expression of these genes to allow drug efflux pumps to be expressed? And if so, if we can understand down to molecular detail how this works, can we find small molecules that interfere with this pathway? So again, just to remind you, this is how it works in humans, and this is how we have shown, as published, it works in yeast and in Candida glabrata. So this is the summary. I'm going to show you the summary first, just so you know where I'm going, and then show you the data supporting the summary. So we found that indeed there is a ligand binding domain in this transcription factor binding to azoles and many different unrelated toxic compounds. That causes a conformational change. This then allows it to bind to this large 30 subunit co-activated complex via a so-called KIX domain, recruit the transcriptional apparatus. These genes will be expressed within 10 minutes of binding of this ligand, and then you efflux the compounds. So this is the way that yeast and Candida can sense antifungals, pump them out immediately. So here's the evidence. So first we showed that if you delete PD-L1 and its paralog PD-L3 in yeast, ligand-dependent activation of this efflux pump is completely abrogated. So you can see that ketoconazole and azole and antifungal can switch on this gene very rapidly within minutes. But if you take out the transcription factor, this guy over here, it no longer works. So that suggests, yes, they can respond to ligands. And PD-L1 actually binds to these compounds. Here's ketoconazole that's labeled with tritium. So it's radio-labeled. It can bind with not too bad affinity. So you have a ligand binding function. And you can actually add cold or unlabeled competitor. And you can see that these actually block the binding of these different, of ketoconazole. So it can bind to many different unrelated chemicals. And so then that allowed us then to map a xenobiotic binding domain in the C-terminus of this transcription factor. So what about, so we found that PDR can interact just like PXR with xenobiotics and respond just like PXR in mammalian cells. What about transcriptional co-activators? Can PDR interact with co-activators? So I mentioned mediator. This is a large transcription co-activator identified in yeast by Roderick Kornberg's lab at Stanford about 25 years ago or so. And what they showed is that it's recruited by transcription factors to DNA, and then it allows binding of RNA polymerase II to genes. And polymerase II can then read the genes, and that then serves as the template. The mRNA serves as the template for protein synthesis. So does PD-L1 recruit the mediator? It does. So we can find here that it interacts with this subunit of mediator and this KIX domain in this pull-down assay. We structurally solved this KIX domain by NMR. So here's the 3-helix bundle showing this structure is very similar here to the mammalian version that we had identified a few years earlier. And we mapped exactly where PD-L1 binds by NMR, showing here, and also by mutagenesis. So it binds to a hydrophobic groove right smack in the middle here. And this was very interesting to us because that suggested that there might be a patch here, hydrophobic patch, where small molecules could nestle in and prevent PD-L1 from interacting. So this was a really motivating finding for us. Perhaps we can drug this pathway. So this is basically what we showed. Just like PXR, PD-L1 interacts with toxic compounds, xenobiotics like azoles, and then bind to this large transcription co-activated complex via this KIX domain. So what happens next? Is this actually functionally important? I just showed you biochemical interaction. So now we take them out genetically, all of these different mediator subunits, we delete them one by one to see what happens. And the only one that has an effect is this GAL11. So if you delete it, the cells are very sensitive to azoles, just like if you delete PD-L1-3 themselves. So that suggests, yes, this is the target subunit. It's functionally important. What if you delete just the KIX domain? Now you no longer see binding of PD-L1 to GAL11. You no longer see ligand-dependent gene activation as compared to wild type and GAL11 full length. So if you delete the KIX domain, it no longer works. And again, just like deleting GAL11 itself, you see that they're very sensitive to azoles again. So that proves that this transcription factor, in response to azoles, causes conformational change. It binds to this KIX domain. This is functionally important to switch on these drug efflux genes to prevent the cells from dying of toxic exposure to azoles. So that's all in Saccharomyces cerevisiae, Baker's yeast. Why do we care? So let's go over to Candida glabrata. It's a cousin of Saccharomyces cerevisiae. They're actually evolutionarily related. And this is a bad bug. As I mentioned, it represents now about 20% of all fungal infections in the clinic. They're intrinsically resistant to azoles. So we wondered, is this pathway we just elucidated in Saccharomyces cerevisiae conserved in Candida glabrata? And we found indeed it is. So they have actually two GAL11s. We deleted both of them. If you delete GAL11A in Candida glabrata, it looks just like deleting PD-L1 in Candida glabrata. They're very sensitive to azoles. They can no longer switch on these drug efflux pumps in response to ligand. So that again suggested we've elucidated a pathway conserved from Saccharomyces cerevisiae to Candida glabrata, whereby this transcription factor can switch on target genes in response to azoles by virtue of binding to this mediator complex to switch on target genes and efflux. Now, can we drug this? So we wondered, maybe we can interfere with this very specific interaction here, perhaps finding small molecules binding to this KIX domain, preventing this from working. So that was the hypothesis. We set out to do this initially biochemically. So we established an in vitro assay where we bind this PD-L1 transactivation domain fluorescently tagged to the KIX domain. And in vitro, we add small molecules, 140,000 completely different chemicals from many libraries. And we can actually prevent this from binding here. And that's the basis for this assay here. So we found about 300 compounds out of 140,000 that are capable of disrupting this interaction. So next, can they get into yeast cells or Candida cells? And so that was the secondary screen. So now we put azoles together with these 300 small molecules, and we're hoping that this small molecule would prevent efflux pumps from being expressed, and then with azoles you would kill the cells. And indeed, we found one compound that could do so out of 140,000. It's this compound, IKIX1. So we show here that in combination of IKIX1 and ketoconazole, you see very efficacious killing of yeast cells and Candida cells. This compound binds directly to the KIX domain, as shown here, and it can prevent the PD-L1 transactivation domain from binding. We structurally solved the complex. First, we solved the Candida glabrata KIX domain. Looks just like the cerevisiae KIX domain. And then we mapped exactly where IKIX1 binds. And it binds in a place that actually overlaps with where the PD-L1 activation domain binds. So here's in red, the PD-L1 activation domain. In blue, this is where IKIX1 interacts. So it makes perfect sense. So IKIX1 binds to the KIX domain, prevents PD-L1 from engaging. Mediator can no longer be recruited, and genes are not switched on. And you get killing of cells because they can no longer efflux the toxic azoles. So first we showed also then that if you add IKIX1 to cells, you cannot recruit mediator complex. This is by using chromatin immunoprecipitation. So you use an antibody binding to the mediator, and you show that it can no longer interact with these target genes, and you can no longer switch them on when you add IKIX1. So this is the expression of the efflux pump. This is, as I mentioned, is conserved in Candida glabrata. So if you add IKIX1, you see in a dose-dependent manner switching off of the target genes. And This happens within minutes after ketoconazole treatment. So ketoconazole switches on this efflux pump expression, as you can see here, but not if you have IKX1. It's completely dead. So what happens to synergy with azoles? So here we show that in wild type Candida glabrata, IKX1 does a little bit, has a small effect on growth, not much. Azole by itself has a small effect by itself, but in combination, they have a synergistic effect. So that was in keeping with the mechanism that we purport, that IKX1 prevents the efflux expression and the azole has an increased efficacy. Now, in Candida glabrata, the main mechanism of drug resistance are mutations in PD-L1 itself. These are mutations scattered throughout the transcription factor that cause them to become constitutively active. So they switch on these drug efflux pumps no matter what. So they don't respond anymore to small molecules, to azoles and other things. You can see here that the wild type is very sensitive to fluconazole and ketoconazole. These mutants are completely resistant. Okay, so can we do anything about that, adding IKX1? And indeed, we find if you combine IKX1 with fluconazole or ketoconazole, you see a great synergy. And even some strains are completely killed by this combination. And we show mechanistically that the drug efflux pump expression, which is constitutive, shown here, there's no, over here, you can see the expression is pretty high. They do respond to some extent to ketoconazole, and that's inhibited, again, in all 4 mutant strains by addition of IKX1. IKX1 also prevents efflux capacity, as you would expect. So if CDR1 and CDR2 are not expressed, you see decreased efflux, which is also what we observe. What about in vivo? Can we have an efficacy on azole-resistant strains in a mouse model? So here we inject this mouse we inject this bug that has this mutation into a tail vein. This is constitutively active Candida glabrata, as you can see here. It's pretty bad. Kidney, spleen, doesn't respond to fluconazole at all, right? Whereas the wild-type strains respond to fluconazole. Now, in the combination with injection of IKX1, we do see a synergistic effect. So about a log or 2 decreased of fungal burden in kidney and spleen. So that's a disseminated infection. Now we also looked in a model for UTI. So in this bladder and kidney assay, we also find that there is synergy between fluconazole and IKX1, especially with the mutant here. And again, supporting the model where this transcription factor is really crucial gatekeeper responding to azoles in the normal circumstances, but can be mutated and become constitutively active and promote this drug efflux pump expression. But we can inhibit this mechanism here and allow accumulation of these toxic azoles in the cells and kill them effectively, in vitro and in vivo. So that was the end of these 2 stories, pretty exciting. We have a small molecule that can actually tackle drug-resistant Candida glabrata, which is rising in the clinic, 20% of patients. How about, can we find better compounds? And so we actually did some medicinal chemistry efforts, and we found compounds that are actually significantly better working together with azoles, with fluconazole here. So we were quite excited about that. Now, we were surprised actually to find, in collaboration with Dominique Sanglard in Switzerland, who's a Candida expert, that IKX1 and analogs actually have intrinsic antifungal activity. Something we had kind of noted before, that there was some efficacy, but not great. There was a great synergy with azoles, not so much by itself, but actually it does have fairly potent activity all by itself. As you can see here, it's about 12 to 25 micrograms per mil in minimal inhibitor concentration. And analogs can have even greater efficacy. And this is actually pan-Candida, across all Candida species. We don't exactly know the mechanism at this point. Why does it do that? We think it's blocking a transcription factor, maybe independent of PDR analogs, something that binds to the Kix domain across all of these different species. And so we're hunting right now for what is that transcription factor that's absolutely necessary for Candida to grow. What about Candida auris? So this is the one that's really scary, that we're caring a lot about right now. So we decided to collaborate with Sean Lockhart at the CDC and Beth Burkov in his lab, and they tested IKX1 on all 100 Candida isolates in the clinic from all 4 clades worldwide. And they actually found that IKX1 has a quite striking IC50, so an MIC of about 4 micrograms per mil. And this is regardless of whether the Candida auris strains are resistant to fluconazole or the last hope, basically, echinocandidates. So it actually can kill all Candida cells regardless of their MDR status. So now we're taking this step further. We solved now the Kix domain of Candida auris, and we show here that it also can bind to PDR1. There must be a transcription factor in Candida auris that's similar to PDR1. We haven't identified it yet, but we're hoping that in the next few months we'll find exactly what that transcription factor is, and we can actually interfere with PDR1 binding to the Candida transcription factor with pretty decent efficacy. And that stimulated us then to try to find even better compounds. So we did additional medicinal chemistry and in silico searches, and we find something that's about 50-fold better than IKX1 in disrupting the interaction between PDR1 and this Kix domain from Candida auris. So we're quite excited. We're hoping that that small molecule inhibitors targeting this pathway in Candida auris might actually be a new avenue, completely new target, unrelated to the standard of care that targets cell wall and membrane biosynthesis. So it actually targets the intrinsic pathway involved in regulating multidrug resistance in these bugs. So again, we're hoping that this will have an impact before this explodes and becomes something like Ebola. But this is very early, of course. Probably have another 5 years or more before we can actually put something into the clinic. So with that, acknowledgements. So this was actually done when I was at Harvard. I was there for 17 years, and I just translocated about a year ago over here to Berkeley. And so these are the folks that did all the work back at Harvard. Collaborators, Hara Arthanari and Andras, and my collaborator, Gerd Wagner's lab. He's done all the NMR spectroscopy structural work, and many other collaborators involved in various aspects of these studies. And a little bit of funding, looking for more, obviously. And thank you so much, and happy to take questions.